Hello everyone and welcome to Nigham's BioNinja. In this video, we are going to cover SLO based MCQs and scenario based short questions with answers from first year biology chapter 1 cell structure and function 1.1 techniques used in cell biology. This is first part of this video series, and in this video we are going to cover the techniques which are cell fractionation and differential staining. These are the techniques which are covered in the textbook. And out of these techniques, these two techniques we are going to cover in today's video, and we are going to discuss the MCQs and scenario based short questions related to these two techniques. There are 20 MCQs and 5 scenario based short questions which are covered in this segment. So without further delay, let's get started with the MCQs. Number 1. Which of the following techniques is used to isolate organelles from a cell? A. Centrifugation, B. Filtration, C. Dialysis, and D. Chromatography. The correct answer is A. Centrifugation. Number 2. Which of the following is not a factor that affects the rate of sedimentation during centrifugation? A. Size of the particle, B. Density of the particle, C. Viscosity of the medium, and D. Color of the particle. The correct answer is D. Color of the particle. Number 3. Which type of centrifugation is used to separate cell components based on their size and density? A. Differential centrifugation, B. Density gradient centrifugation, C. Preparative centrifugation, D. Analytical centrifugation. So the correct answer is B. Density gradient centrifugation. Number 4. Which of the following is not a common method of disrupting cells during cell fractionation? A. Sonication, B. Homogenization, C. Enzymatic digestion, D. Osmotic lysis. So the correct answer is C. Enzymatic digestion. Number 5. Which of the following organelles is usually the first to sediment during differential centrifugation? A. Mitochondria, B. Nuclei, C. Ribosomes, D. Lysosomes. So the correct answer is B. Nuclei. Number 6. Which of the following is a disadvantage of using differential centrifugation to isolate cell components? A. It requires expensive equipment, B. It is time consuming, C. It is not very precise, or D. It can damage delicate organelles. So the correct answer is B. It is. Number 7. Which type of cell fractionation method uses a sucrose gradient to separate cell components? A. Differential centrifugation, B. Density gradient centrifugation, C. Preparative centrifugation, D. Analytical centrifugation. The correct answer is B. Density gradient centrifugation. Number 8. Which of the following is not an advantage of using cell fractionation techniques to study cell components? A. They allow for the isolation of individual organelles or cell components for detailed analysis. B. They are relatively quick and easy to perform. C. They provide a way to study the biochemical properties of organelles in vitro. D. They can help identify specific proteins and their functions within organelles. The correct answer is B. They are relatively quick and easy to perform. Number 9. Which type of centrifugation is used to separate proteins based on their size and shape? A. Differential centrifugation, B. Density gradient centrifugation, C. Preparative centrifugation, D. Analytical centrifugation. So the correct answer is D. Analytical centrifugation. Number 10. Which of the following is not a common application of cell fractionation techniques? A. Study the biochemistry and function of organelles, B. Identifying specific proteins within a cell, C. Diagnosing diseases at a cellular level, or D. Producing recombinant proteins for medical use. The correct answer is C. Diagnosing diseases at the cellular level. Number 11. Which of the following dyes is commonly used to stain DNA in eukaryotic cells? A. Methylene blue, B. Hematoxylin, C. Eosin, D. Safranin. So the correct answer is B. Hematoxylin. Number 12. Which of the following is an advantage of using differential staining techniques to study cells? A. They can be performed quickly, B. They allow for the visualization of cellular structures and components, C. They do not require expensive equipment, D. They do not require specialized knowledge or training. So the correct answer is B. They allow for the visualization of cellular structures and components. Number 13. Which of the following stains is commonly used to stain bacterial cells? A. Methylene blue, B. Hematoxylin, C. Eosin, D. Safranin. So the correct answer is D. Safranin. Number 14. Which of the following structures in a eukaryotic cell is stained with the acid-fast staining technique? A. nucleus, B. cell membrane, C. mitochondria, D. cell wall. The correct answer is D. cell wall. Number 15. Which of the following stains is commonly used to visualize the Golgi apparatus in eukaryotic cells? A. hematoxylin, B. eosin, C. toluidine blue, D. periodic acid shift, PAS. So the correct answer is D. periodic acid shift, PAS. Number 16. Which of the following dyes is used to stain the cytoplasm of cells in a Gram stain? A. crystal violet, B. safranin, C. iodine, D. malachite green. So the correct answer is B. safranin. Number 17. Which of the following types of cells is not effectively stained by Gram staining? A. bacteria with a thick cell wall, B. bacteria with a thin cell wall, C. archaea, or D. fungi. So the correct answer is C. archaea. Number 18. Which of the following is a disadvantage of using differential staining techniques to study cells? A. They are not very specific, B. They can be time consuming, C. They can damage cells, D. They are expensive. The correct answer is B. They can be time consuming. Number 19. Which of the following is a common application of differential staining techniques? A. diagnosing diseases, B. studying the structures and functions of cells, C. producing recombinant proteins, D. purifying proteins from cells. So the correct answer is B. studying the structure and function of cells. Number 20. Which of the following stains is used to differentiate between bacterial endospores and vegetative cells? A. Gram stain, B. acid-fast stain, C. endospore stain, D. Ziehl-Neelsen stain. So the correct answer is C. endospore stain. That was all about MCQs from these two topics. Now we will switch to SLO based or scenario based short questions. Number 1. Aisha is studying the structure of the plasma membrane of a cell. What technique can she use to isolate the plasma membrane? The answer to this will be Aisha can use differential centrifugation followed by ultracentrifugation to isolate the plasma membrane from the rest of the cell. The cell is first homogenized and the resulting homogenate is centrifuged at a low speed to remove the nuclei and unbroken cells. The supernatant is then centrifuged at a higher speed to obtain a pallet containing the microsomal fraction which contains the plasma membrane. The microsomal fraction is then subjected to ultracentrifugation on a sucrose gradient to isolate the plasma membrane. Number 2. Saad is studying the RNA present in the nucleolus of a cell. What techniques can he use to isolate the nucleolus? The answer to this will be Saad can use cell fractionation followed by ultracentrifugation to isolate the nucleolus from the rest of the cell. The cell is first homogenized to obtain a crude fraction that contains all cellular components. The fraction is then subjected to ultracentrifugation on a sucrose gradient. The nucleolus will sediment at a specific density on the gradient and can be collected and analyzed. Number 3. Zainab is studying the chloroplast of a plant cell. What technique can she use to isolate the chloroplasts? The answer to this will be Zainab can use differential centrifugation to isolate the chloroplast from the rest of the cell. The chloroplasts have a higher density compared to other cellular components and can be separated from the rest of the cell by differential centrifugation. The cell is first homogenized and the resulting homogenate is centrifuged at a low speed to remove the nuclei and unbroken cells. The supernatant is then centrifuged at a higher speed to obtain a pallet containing the chloroplasts. Number 4. Fatma is studying the tissue samples of a patient to diagnose cancer. What differential staining technique can she use? The answer to this will be Fatma can use the hematoxylin and eosin H and E staining technique to differentiate between different cell types in a tissue samples. Hematoxylin stains the nuclei blue, while eosin stains the cytoplasm and extracellular matrix pink. This technique can help identify abnormal cell types which may indicate the presence of cancer. Number 5. Ali wants to differentiate between gram-positive and gram-negative bacteria. What differential staining technique can he use? The answer to this will be Ali can use the gram staining technique to differentiate between gram-positive and gram-negative bacteria. In this technique, the bacteria are first stained with crystal violet followed by iodine solution. The slide is then washed with alcohol and counterstained with safranin. Gram-positive bacteria will retain the crystal violet stain, while gram-negative bacteria will lose the crystal violet stain and take up the safranin counter stain. That's all for today from these two topics. I hope that you have learned enough. For the remaining techniques, please watch the remaining 3 videos as well, and don't forget to subscribe to my channel for further information. Thanks. Until next time, this is Nigham's BioNinja signing off.